Okay, welcome to week 2, and in this week we're going to learn about DNA and DNA replication. So at the top you can see the word replication. There's also another word that means replication, and you're going to see it a lot, and it's called the word synthesis. Synthesis just means to make, or in this case to replicate. So what we're doing in this week is we're learning about the replication or the making of new DNA. And we need that in all of our cells. So if you take a look at this picture, this is the picture of what DNA actually looks like. We have DNA in all of the cells in our body, and it takes the shape of this picture. This picture is showing the shape of DNA, and it's called a double helix. So it looks like a winding staircase, and it's just molecules twisted together, and this is the genetic makeup of anyone and everyone and every living thing. All living things have some form of DNA or RNA. So if you take a look at this picture, this is a picture, and all of the organisms in this picture are humans. They're all Homo sapiens. But what's different about them is all of their physical traits. Every single person in this picture looks different in some way. The DNA in humans is very similar, but there are some slight differences that make us look very different from one another. And that's the really cool thing about DNA, is our DNA is very similar, but the slightest changes can make us who we are. Alright, so in this slide I want to show you just a little bit about DNA and what makes up DNA. So there are several things that make up DNA, and what we're going to focus on is the actual nucleotides that make up the DNA and are considered our genetic code. So the nucleotides in DNA are represented with letters. Okay? A stands for the nucleotide adenine. T stands for the nucleotide thymine. G stands for the nucleotide guanine. And C stands for the nucleotide cytosine. Okay, these all have a relationship with each other, and when we talk about DNA, it's really important that you remember the sequence that I'm fixing to tell you. Okay, the next thing that I want you to remember is I want you to write this down in your notes that you've downloaded for this week. So when we look at this DNA, we see that in this strand of DNA we have an A that is connected to a T, a G that is connected to a C, and vice versa. So the relationship with DNA is it's a double helix. One side of the DNA is complementary to the other. What does that word complementary mean? That means it is the same, but it has opposite parts. I know that sounds a little confusing, but this is one way you can remember what bonds with what. So there's a little acronym that I like to remember. At the Gulf Coast. We live really close to the Gulf of Mexico, all right? And this acronym helps me remember what side of this DNA can bond to this side. So every time adenine will bond to thymine. So A bonds with T, and G bonds with C. So this acronym helps me remember at the Gulf Coast. A bonds with T, G bonds with C. So these are the complementary base pairs or complementary nucleotides that combine with each other. So A bonds with T, A bonds with T, G bonds with C, C bonds with G, C bonds with G. Okay, so if I were to put another letter here, what would be on the other side? Pause the video and see if you can answer this. The answer is A. Okay, So all you have to do is memorize this little acronym, and I think you'll be fine for this section of the slide. Okay, so now we're going to get to the nitty-gritty of when we look at DNA replication. So what we have is the DNA strand, okay, and DNA is always double-stranded. We have an enzyme, okay, and the name of that enzyme is called helicase. All right, so the job of helicase is to unzip or unwind that double helix and break the bond between the complementary base pairs. Okay, so why would it need to do that? Well, if we're going to duplicate the DNA, we need to separate that double strand of DNA. Okay, so that is the job of helicase. Helicase is a protein that comes in and unzips the DNA and exposes the base pairs and the nucleotides to another protein that we're going to talk about next. So this would be the first step of DNA replication. So I would make sure that I wrote that, kind of like a summary of that in my notes, and also you'll need that for your outline. Alright, and one thing that I want to mention is there's a bond that kind of holds these nucleotides together, these complementary ones. Okay, the complementary ones are held together by a hydrogen bond. So that's what this green dotted line represents. It represents a hydrogen bond. Alright, the next step is when we actually read the DNA strand and we make an exact copy of the DNA. Okay, I know there's a lot going on in this picture, but just bear with me and I think you can figure it out. So after DNA helicase comes in and unzips the DNA, there's another enzyme that comes in and it's called DNA polymerase III. The job of DNA polymerase III is to make more DNA or make an exact copy of DNA. This is called elongation. Okay, so let's pretend that DNA polymerase starts right here. Okay, what it does is it's going to read this bottom strand and it's going to take in a nucleotide from the outside cytoplasm somewhere, and it's going to match up with its complement base pair. So what did we say bonds with C? G. Okay, and then it goes along and it reads this one. It recognizes a C. It's going to add another G. All right, then it will come along and it will recognize a G, and it will add a C next. So let's kind of make this work. There we go. So it would add a C. Then it would continue on, and then it would add the next letter, which would be a T because it recognized an A, and then the last one it would add would be an A. Okay, this process happens very quickly, all right, and this DNA polymerase can read hundreds and hundreds of DNA of base pairs per second. So this is a very fast, and DNA polymerase is really good, and it hardly ever makes a mistake. One thing that limits DNA polymerase, though, is the direction it can go. All right. If you notice, I have a 5' here and a 3' here. That represents where the nucleotide is attached to the phospholipid backbone, or the phosphate backbone of DNA, which would be represented by this blue line. So the 5-carbon part of the backbone is connected to the C on this end. Toward this end, it's connected to the 3'. So as you go from 5' to 3', that is the only direction that DNA polymerase can travel. So that is a little bit cumbersome, because on the other side of DNA is opposite. So if this is 5', this is going to be 3'. So what it has to do is it has to work backwards, okay? And instead of elongating in one big swoop, it can only go a little short ways, and then it has to stop on the second strand. Okay, the second strand is not represented here to make it a little more simple and easy to see. Okay, so what do I want you to remember about this slide? I want you to remember that elongation is where DNA is copied by DNA polymerase and added to a new exact copy DNA strand. Remember, DNA polymerase can only go in one direction, 5' to 3'. Okay, And on the next strand, or the other strand, that's called the lagging strand. It can only go in segments. In the leading strand, which is what this shows, it can continuously go without having to stop. Alright, so let's do a little review. So what we talked about is DNA replication, and we talked about a couple enzymes. Okay? If you remember back from when we talked about macromolecules, we learned that DNA polymerase, or anything that ended in the three letters ACE, A-S-E, was going to be a certain macromolecule. Pause the video and think about which macromolecule you think DNA polymerase might be. Okay, so let's see if you got it right. So here is our answer. Okay, DNA polymerase is an enzyme, so it has to be a protein. Any enzyme that we talk about is going to be a protein. That's the macromolecule. All right, what is DNA polymerase's job? Is to read the DNA strand and add the complements to a new DNA strand to make more DNA. All right. What has to happen first, though, in the very first step of DNA replication? Well, we have to have an enzyme that comes in and it has to unzip the DNA. Pause the video and see if you can remember the name of this enzyme. Okay, so what did you come up with? This enzyme that unzips the DNA is called helicase. Okay, well, I hope you've taken good notes. Please make sure you email me with any questions about this. Also, remember you have the outline and the notes, and also you have the script that you're going to be writing for this section. Okay, go back and review the instructions if you have questions about that.